Hey, everybody. Welcome to the MaxGen Labs Practitioner of the Month series. I'm Chris Grout, and our speaker today is Brendan Vermeire. Today's topic is COMT, methylation, and mast cells, unraveling genetic links to mental health, inflammation, and women's health. Brendan is a board-certified holistic health practitioner, master nutrition coach, master personal trainer, USAW sports performance coach, and CrossFit trainer. He's also a functional medicine educator, mental and metabolic health scientist, researcher, writer, and speaker. So lucky to have you here today. So glad that you're here. And remember, everybody, if you have any questions that you might have during the presentation, put them in the chat and they'll be addressed at the end of the presentation. With no further ado, welcome, Brendan.
Thank you so much for having me, Chris. It really means a lot. I love MaxGen and the work that you guys are doing. So it's always fun anytime we can share some cool information. So thanks for having me.
Absolutely. I'm going to check out now and let you take over.
Sounds good, brother. Thank you. There we go. All right. I hope everybody's having a great day so far. I really appreciate the fact that you guys would take time out of your busy weeks on a Wednesday to come hang out, learn about genetics, epigenetics, nutrigenomics, neuroinflammation, all the cool stuff that we're going to talk about today. I love getting to put together really cool clinical education content, try to advance the field. And certainly, I get really excited to work with MaxGen because certainly I think genomics and genomic precision medicine, that is the future. So I look at MaxGen as a group that is contributing to the future of medicine. Now, certainly we have a long ways to go because when we really look at the human genome there's at this point in time estimated to be about 23,000 protein coding genes in the human genome. Every single one of us bipedal hominid humans, we all have 99.9% genetically identical, but it is that 0.01%, the single nucleotide polymorphisms or SNPs that make us each unique of how we look, how we sound, how we behave, how we think, how our mental health is. So I think looking at SNP analysis and looking at the genetic predisposition and then looking at all of the different lifestyle, environmental, and even psychosomatic input signals that are going to alter and modulate our epigenetic expression, our protein expression. I always like to say that thoughts become proteins, very much our mindset, our chosen beliefs, our chosen actions. This does actually influence our epigenetic health and therefore physiology. Now, at this point in time, it's estimated that there's about 88 million single nucleotide polymorphisms that have been identified in the human genome. Now, they're discovering new SNPs all the time. The question is, which of these SNPs are really clinically relevant? As in, if you have one of these SNPs, how much could it possibly alter the outcome of your life, your clinical outcomes, your health outcomes? So certainly when we think of some of the more clinically established, clinically relevant polymorphisms, it's a much shorter list. So I think we do have to practice a certain degree of humility and curiosity in regards to there's probably a lot of naughty polymorphisms that we just really haven't discovered yet. And it's really impossible to predict somebody's health outcome just based on the genetic predispositions. And so what I wanted to try to accomplish in this presentation is taking a deeper look specifically at the COMT polymorphism and then how this relates to clinically relevant, actionable methylation biochemistry, neuroimmunology, the state of inflammation, hormonal recycling, gut-brain axis, really tying in the genetic blueprint and predisposition with having a fast or slow or mixed COMT, and how this can then influence very real clinical outcomes in practice. So without further ado, let's go ahead and dive in here. Skip ahead. You guys can check this out if you're not familiar with me or my work. But these are our learning objectives for today. I think this presentation, it turned out relatively dense. I had a lot of fun building it. I've certainly done a lot of work around MTHFR. The last time I did a webinar for MaxGen, it was more focused on MTHFR, methylation, and nitric oxide, which was a really cool presentation that I'm sure you can catch the recording of somewhere else. But now I want to expand the conversation, move forward, take a closer look at COMT, because I think we've all probably seen, if you're tuning into this live webinar right here, right now, you are very advanced with your genetic understanding. And I think that all of us who are genetic enthusiasts, we've seen how MTHFR has really blown up, right? Now, MTHFR isn't anything new, to give credit where credit's due. I mean, Dr. Ben Lynch was rocking the MTHFR, beating the drum 14 plus years ago. And certainly there's other predecessors before him. But we're seeing this kind of mass level of awareness around, well, MTHFR, and it's unpacking this whole, well, what does MTHFR mean? And what do I do about it? And what's this methylation thing? So, it's probably just a matter of time before now everybody's kind of interested or freaking out about the COMT or ApoE or whatever the enzyme in question is. And certainly as epigenetic nutrigenomic practitioners, we have a more advanced understanding of how, hey, well, if we have these sort of biochemical predispositions that are creating a weakness within a certain biochemical pathway, how could that potentially alter our physiology? And what biomarkers and tests can we really use to objectively evaluate, is this SNP that I see in my MaxGen report actually impacting the clinical outcome, and what can I do about it to course correct, right? So of course that blueprint will inform us of the potential possibilities of what might be plaguing them, or why do they continue to struggle with certain symptoms, or why are certain biomarkers like homocysteine always a little bit out of range, right? So we can get those clues and then better be able to better tailor their bio-individualized protocol to move through some of those healing roadblocks that we all encounter along the way. So we'll take a look at, obviously, the basics on COMT. What is it? How does it relate to neurotransmitter metabolism specifically, and then therefore concomitantly mental health? We'll learn how to differentiate between more of a fast COMT versus slow COMT, and looking at the specific SNPs that can predispose somebody to more of a faster COMT activity versus a more kind of sluggish, slow COMT activity, and then starting to explore some of the fun interconnected mechanisms of what are the potential clinical implications if we have fast or slow COMT. So we'll add some contextual clues and different layers, different types of scenarios like estrogen dominance that many of us are seeing in our practices literally every single day. And then how do we then tie in that MTHFR methylation component, right? We all know, or I would imagine many of us know, that catechol low methyltransferase being a methyltransferase enzyme. Well, that means that the ability to produce methyl donors, specifically S-adenosyl methionine, or superhero SAM for anybody that's heard me lecture about methylation before. Well, we have to have plenty of methyl donors to be able to even activate COMT and use it to break down estrogen or catecholamines or whatever else we're trying to metabolize. So looking at how the COMT, not just the SNP, but the actual functional enzyme, can relate to gut health, inflammation, estrogen dominance, and mast cell activation syndrome, neuroinflammation, so much more. And how can we use genomic precision medicine here in conjunction with other functional lab tests and investigations? So it's quite the docket. We'll see how we do with it. And please don't be shy. Be sure to drop your questions and comments into the chat box. I'll probably, for the sake of my COMT activity and my ADHD, I'll try to stay focused on the presentation, but I want to make sure that we leave at least a little bit of time to address some live questions, because those are a lot of fun. Now, this was actually just posted on Instagram the other day from MaxGen, and I collaborated on that post. This is really, I guess, my working theory for my whole career. Based on what I think I know, I will very likely spend the rest of my life really pioneering this work. And I even had somebody kind of rebuttaling this on Instagram. And it's like, I don't think they understood what I really meant with this statement. So, I would argue that mental illness is largely, I say, I'm very careful with my verbiage, largely, not exclusively, but largely an epigenetic phenomenon driven by oxidative stress, inflammation, and neuroendocrine dysregulation, right? Now, I use that word epigenetic very strategically, because while we all are going to have some naughty single nucleotide polymorphisms and SNPs and genetic mutations, we really want to use our environmental input signals, our lifestyle and lifestyle behavior input signals, our chosen beliefs, our supplements, medications, other therapeutic modalities to modulate the epigenetics and really get our health to where we want to be. And so I look at this as how do we improve the level of functionality within this particular individual, regardless of whatever their predispositions are. But by knowing their SNPs and knowing their predispositions, their genetic strengths and weaknesses, we can more further accurately tailor their protocol to their specific needs. So as far as some just basics on COMT, or catechol-low-methyltransferase. So as we discussed, it's a methyltransferase enzyme that's going to use S-adenosyl methionine as the body's major methyl donor to essentially methylate some sort of molecule to metabolize it through phase 2 detoxification methylation reactions here. Now, COMT plays an absolutely critical role, particularly within the central nervous system, as well as the peripheral nervous system in general. But we actually have two different types of COMT, and this never really gets talked about. But there's going to be the soluble COMT, which is primarily in the liver and the kidneys and peripheral tissues, versus the membrane-bound COMT. This is going to be more so relevant for our conversation about neurochemistry within the brain. So we have this presynaptic neuron, postsynaptic neuron here in the diagram, showing the conversion of tyrosine into dopamine. It's obviously skipping a lot of biochemical steps there. We package it up into a vesicle, and then when we have a nerve impulse, we will release that stored dopamine or whatever neurotransmitter it is into the synaptic cleft via exocytosis. So that way we're basically dumping those molecules of neurotransmitter into the synaptic cleft, where they should bind on to corresponding receptors on the postsynaptic neuron to convey that nerve impulse and move things along in the central nervous system to convey these signals. Now, obviously, the activity within the synaptic cleft is largely going to influence our neurotransmission and therefore our cognitive capacity, and also therefore our mental health, our psychological state, and so much more. So when we're talking about this catechol-low-methyltransferase, its job is to really break down our catecholamines, so our dopamine, norepinephrine, and epinephrine in particular. And there are other enzymes that are involved in both the biosynthesis and the metabolism, such as MAO, or dopamine beta hydroxylase. So there are other SNPs that maybe we'll do future webinars for to even, further expand that conversation. But if we can think about the importance of being able to not only produce adequate levels of dopamine and serotonin and GABA, glutamate, acetylcholine, so on and so forth, but especially with this catecholamine conversation, we all think about dopamine as pleasure and reward and focus and our ability to execute tasks, right? So being able to have adequate dopaminergic signaling in different parts of the brain. So for example, psychosis or different psychotic disorders such as schizophrenia are largely related, not exclusively, but largely related to a dysregulation of dopaminergic activity in different brain regions. Some brain regions, we have too much dopamine signaling, which is why various antipsychotic medications work by blocking the D2 dopamine receptor, for example, whereas maybe in other brain regions, there's actually too low or sluggish of dopamine activity that's causing the apathy, the anxiety, and the depression, right? So certainly looking at, well, what's going on with their COMT enzyme based on the genetic predisposition, and are they very quickly breaking down their dopamine? Are they not breaking down catecholamines fast enough, and so they're really anxious and stressed out? It totally depends, but by informing our clients and patients of their genetic predisposition, we might get answers that other physiological biomarkers don't really give us the same degree of insight for. So if we have any kind of variation, and there is more than one SNP for COMT, but we're going to be focusing on the best studied single nucleotide polymorphism with COMT. But essentially, the functional capacity, the functionality of the COMT enzyme that's coded for by the COMT gene is really going to influence the dopamine activity, our mood, our cognition, and even things like pain perception. So now this opens up the conversation on fast or slow. I see this all over, at least my social media, because this is, stuff I talk about and post about relatively regularly. So a lot of people are trying to figure out, well, am I a fast COMT or a slow COMT, and what do I do about that? So specifically, we're talking about the Val 158 MET SNP. RSID is 4680 if you want to look up the specific gene. Now, like I said, I do have a case study that I want to try to get to, and I have an example of a MaxGen report where we can see multiple COMT SNPs. The Val 158 MET is the best studied, and at this point in time, it is considered the most clinically significant in relation to the fact that if this SNP is present, it more profoundly alters the functional capacity of the COMT enzyme that then is going to influence our biochemistry and especially our neurochemistry. So if we have a val-val, this is valine-valine. So this is a fast COMT. This valine-valine would essentially mean that our COMT enzyme is operating at a more robust, fast level, which means that we're going to break down our, I was going to say feel-good catecholamines, but they don't always feel good. And then I wanted to say our stressful feeling catecholamines like norepinephrine and epinephrine. But I mean, both can be true at the same time. We need a balance of catecholaminergic signaling to be able to respond to stressors or stimuli. I don't know about you all, but I find with my day-to-day life and everything, if I don't have enough to do, like my workload isn't enough, if I don't have enough to kind of rev me up, there's eustress and distress. We actually need some degree of eustress to keep us motivated, to keep us going, to stimulate our neurochemical systems, to have drive and execute tasks and do stuff, right? But if it's too much, then obviously that can fall into distress, which can obviously lead to like an HPA axis dysfunction. We start feeling burnout and stressed out and chronic fatigue and all of these things, right? So there's a myriad of genes and genetic polymorphisms that can influence our ability to be plastic, to bounce back from different stressful input signals. As functional practitioners, so much of what we do is try to remove as many root cause hidden stressors that are contributing to this allostatic burden, this allostatic dysregulation that moves us away from a healthy, happy homeostasis and into dysregulation, dysfunction, and possibly even disease. So with a fast COMT, and then we have the slow COMT, which is the methionine, or just MET-MET. So this is more our slow COMT. It's a sluggish COMT. This is kind of comparable to the homozygous 677 MTHFR polymorphism, for those of you that are familiar, meaning that the slow COMT is going to be operating much more slowly. So some of these catecholamines, dopamine, norepinephrine, and epinephrine, can start building up. They're not being metabolized as quickly. So, you know, I remember over the years, I've done a lot of clinical advising with a lot of practitioners. And so I always like to use different analogies to teach a concept or a point, because even on, let's say, like the Dutch test, you know, urinary hormone analysis, I think a lot of you know what I'm talking about with the estrogen metabolism. They actually have a little gauge that's showing, you know, your 2-hydroxyestrone to 2-methoxyestrone. And essentially it's estimating the ability of your COMT enzyme to methylate or metabolize that 2-OH-E1 into 2-methoxy-E1, and it gives you that little gauge. Now, that's not really an accurate reflection of COMT activity. It's more looking at the ability of COMT to methylate the estrogen radical or metabolite into a conjugated, you know, form that then we can excrete. But so when we consider, well, how much of the substrate is being biosynthesized and produced, and how much of it is breaking down? So my good friend, Dr. Carrie Jones, giving credit where credit's due, you know, she always uses the bathtub analogy with estrogen dominance, right? So if you think about like how much water is flowing into the bathtub and filling it up, you know, that's the faucet. Is it on full blast and you're biosynthesizing a ton of estrogen or a ton of dopamine or whatever the substrate in question is? And so how full is that bathtub? But then how wide open or closed is the drain, right? Because we really have to think on both sides. Same thing with histamine as well, which we're going to talk about a bit today. So how much biosynthesis of that molecule, hormone, neurotransmitter, metabolite, versus how quickly can we break it down and excrete it. So using that bathtub analogy for our catecholamines, if we have a slow COMT, MET-MET, it's going to be a harder process to drain the bathtub. And so we get this sort of overflow of dopamine and catecholamines that could lead to symptoms like rumination, for example. So the case study that I'll show you guys in a little bit is actually an OCD case study. And one of the major symptoms of obsessive compulsive disorder is this chronic rumination. You know, people get really stuck on this sort of circular thought loop in a negative way. It's like burning a hole in their mind. They can't let go. They're ruminating excessively, and it's sort of keeping the stress alive, keeping the trauma alive. And not that we can blame it all on a single SNP, but certainly if they have a slow COMT, and let's say they're pumping a bunch of, tyrosine or L-DOPA, methylated B vitamins, so they're pushing a lot of nutrients that produce the catecholamines, but meanwhile they have a slow COMT, so they can't really drain the bathtub very quickly. Well, it's going to add to more stress and anxiety and rumination and exacerbate their mental health disorders. So we could certainly fill in the blank with other scenarios, ADHD or bipolar, whatever the case is, but we're really looking at some of the underlying mechanisms that could be contributing to the clinical presentation and symptomatology. Now, the case study I'll show you, she is I believe she's a slow COMT. We'll take a look at that. I checked my MaxGen report right before this webinar because I couldn't remember. I do know that I have ADHD and I'm trying to stay focused and on track here, but my Val 158 MET SNP, I'm actually pretty balanced. I'm an intermediate COMT. So, my ability to manage my own ADHD is largely influenced by, how I'm supplementing and supporting these particular pathways. I supplement with a dopamine formulation every morning. I pop it with a methyl B complex to push those nutrients through this pathway. And, certainly if I overdo it, I have a moderate enzyme capacity with COMT, but if I push those nutrients too much too hard, yeah, I can get a little bit strung out myself. And certainly there's a lot of other variables that we have to be considering as well. So I think I've made the point with the analogy. So moving forward here. So some of the clinical implications that we might want to be looking at, depending on when we look at that MaxGen report, are they the Val fast COMT? Are they a MET slow COMT? Or are they intermediate like me? So individuals with a fast COMT, this does not mean they're automatically going to be experiencing these symptoms. It simply means they're going to be more predisposed to experiencing these symptoms more often, more likely than somebody that has a different genetic predisposition. So fast COMT would predispose us to more anxiety, difficulty with sustained attention, impulsivity, and higher pain threshold. I don't know about you all, maybe it's because I was diagnosed with ADHD when I was 21, and I kind of wonder, like, had I known earlier in life, could that have changed my academic performance and some of my lifestyle choices to better cater to my individual needs? So I think of like all the young people, all the children, especially the young boys that have ADD, ADHD, being asked to sit still for 8 hours at school and study stuff that they don't care about. So certainly I just think about all the maybe kiddos that, like, gosh, it's really easy to get a saliva sample, test their genetics, and then we can just further support them in their life path at an earlier age. That way we don't have to, undo a bunch of damage and dismantle a bunch of self-limiting beliefs, later in life and adulthood. But so if we have more of and also a higher pain threshold, which I think is kind of an interesting clinical pearl that I wanted to highlight, but also impulsivity. I think all of us can be a little bit impulsive at times, but if you're a fast COMT, you might be a little bit more impulsive than others. Whereas a slow COMT, okay, these individuals are perhaps going to have a better working memory, but they're also going to have more obsessive thoughts, potentially maybe more rumination, can't really let things go as much, possibly. Now, because of some of the physiological interplay and mechanistic overlap, they could also then have higher estrogen levels. One of the major functions of COMT, as I already have alluded to, is estrogen metabolism. And so certainly we could have a whole conversation about the prevalence of estrogen dominance and all of the negative ramifications and clinical implications of estrogen dominance. But because COMT is a major enzyme to help us break down and methylate our phase 1 hydroxylated estrogens, we have to be able to clear those out of the body. And I'll elucidate that more as we go because we have some subsequent slides. So moving forward here, factors that modify COMT activity. Now, this is absolutely critical, so I'm going to kind of take my time here because, again, we can't really assume what their physiology is doing and what symptoms are attributed to what just based off the genetic blueprint. That's just informing us. It's giving some clues because then, with that blueprint, okay, maybe you have a coffee stain on the blueprint and so it's kind of smudged and makes for a faulty, less functional protein enzyme that actually we need to facilitate some sort of biochemical reaction, right? So other factors that are going to actually alter the epigenetic expression of the protein enzyme from the blueprint gene. Well, looking at estrogen, this is kind of cool, kind of interesting. So estrogen actually downregulates the expression of the COMT enzyme from the gene, which you think about this for a second, it's like, well, wait. So we need plenty of functional COMT protein enzymes to be able to break down estrogen. So if we have a slow COMT, that could predispose us to increasing levels of estrogen. But then as estrogen is increasing in concentration, it's actually down-regulating the production of the COMT protein enzyme. So if that made sense and you followed that, you can imagine how this could become a vicious cycle, right? Because as estrogen starts building up in the body and we get more and more estrogen dominant, it's actually then decreasing the expression of the enzyme that we need to be able to break down that estrogen to begin with. So it's kind of like a estrogen dominance fuels more estrogen dominance in part because it's down regulating the expression of the enzyme that we need to break it down, right? Now, further, with methylation, of course, we have to have either functional MTHFR enzyme or, well, we have to bypass the enzyme using methylated B vitamins to be able to produce adequate levels of S-adenosylmethionine or SAMe, along with BH4 or biopterin in order to produce these neurotransmitters and the methyl donors needed to facilitate this biochemistry. So we'll take a closer look at that with methylation because that gets a little bit technical and I have some graphics that'll help illustrate that point. Now, further, with inflammation, this is something I'm super into. There's a lot of different noxious mediators, things like free radicals and reactive oxygen species, things like, different medications or even botanical phytonutrients, or specifically more the noxious mediators like lipopolysaccharide from leaky dysbiotic gut, mycotoxins, heavy metals, acetaldehyde, different pro-inflammatory cytokines such as interleukin-6 and TNF-alpha. Some of these different noxious pro-inflammatory sort of pro-disease mediators can also negatively impact the epigenetic expression of the enzyme from the blueprint gene. So for example, interleukin-6, which we all know is kind of the most popular, the best studied, the most talked about pro-inflammatory cytokine, and then TNF-alpha, interferon gamma, or interleukin-1 beta, or some of the others there. But interleukin-6, which any of us that are using blood work, which I hope is all of us, to routinely track our client patient's physiology, good old-fashioned C-reactive protein. There's better inflammatory markers that exist now, but good old-fashioned C-reactive protein is very reflective of interleukin-6. So if you have somebody that's walking around with a C-reactive protein of 3 on the low side, 7 or 8 on the higher side for kind of that low-grade chronic inflammation sequelae that so many of us are dealing with in our client patient population, well, even that high CRP should be informing us of like, well, clearly they have some degree of inflammation, upregulated interleukin-6, and that could be then downregulating the expression of the COMT that we need to effectively clear our catecholamines and our estrogen, so on and so forth. So as you can see here, there's so many different mechanisms and pathways that we could really focus on. I'm just trying to give you a conceptualization of how to approach this in clinical practice. And then obviously, what biomarkers or lab tests can we use to corroborate our clinical critical thinking, so we have actionable data that then we can track over time to make sure our theory worked, our protocol worked, and we have the objective data that we can prove it. We don't just have to go based on subjective symptomatology that's going to be all over the board, all over the map. So taking a little bit closer of a look, I really like this diagram and graphic, by the way. I thought it was kind of fun, kind of cute. So looking at the role of COMT in mental health, dopamine, mood, and cognition, COMT regulates dopamine in the prefrontal cortex. So this is going to directly impact our ability to focus, decision-making, emotional regulation. I don't know about you all, but I know a few people that probably could use a little bit more capacity for emotional regulation, a little bit more equanimity and stoicism. I wonder if Marcus Aurelius had a balanced COMT, or if he was fast or slow. I don't know. Seems like a pretty even-keeled dude from what I've read. Now, with a fast COMT, that's going to predispose us to lower dopamine levels. This could then obviously lead to more anxiety, lower stress tolerance, more impulsivity. That slow COMT that might predispose to higher dopaminergic activity, overthinking, OCD tendencies, difficulty switching tasks. Whereas we really want balance, because that low dopamine is going to be a higher risk of ADHD, anxiety, depression. The high dopamine, greater cognitive persistence, the ability to do a lot of cognitive load and workload, but increased stress sensitivity, maybe a little bit more snappy, a little bit more kind of pent up, about to burst, if you will. So this allows us to then further individualize their particular protocol or program that we're coaching them through. Now, to add in another layer here, so certainly genetics and methylation and neurochemistry, big areas of passion for me. The gut-brain axis, the microbiome. I actually am on the clinical team at Microbiome Lab, so it's impossible for me to not bring in the microbiome to every single lecture that I do. And this is actually pretty critical, because when we think about the role of the estrobolome, so those of you that are microbiome savvy practitioners, obviously, with the human microbiota, we have a lot of different components of the microbiota. We have the bacterial component, the bacteriome, the fungal component, the mycobiome. We have the virome, or the viral component, the parasite component, but we also have the psychobiome, the component, the bacterial species that produce neurotransmitters, such as Klebsiella, which is often thought of as an opportunistic pathogen associated with things like rheumatoid arthritis and Hashimoto's and Sjogren's. Well, Klebsiella is a gram-negative bacteria. It's opportunistic. It can release lipopolysaccharide endotoxin that can fuel that interleukin-6 and macrophage activation and inflammation in the body and the brain. But also, Klebsiella produces histamine itself directly and can contribute to excess histamine in the gut, the body, the brain, and really overlapping with mast cell activation. The lipopolysaccharide that it releases could potentially activate mast cells within the gut that are then degranulating and releasing a lot of histamine and tryptase and heparin and serotonin. This can damage our intestinal lining, causing more leaky gut, more endotoxemia, more translocation of these pathogen-associated molecular patterns that stimulate a lot of inflammation in the gut, the body, and potentially the brain if it makes it there. But then it's also potentially contributing to the histamine burden as well. So there's so much crosstalk and cross mechanisms going on here. But with the estrobolome, this is the component of the microbiome that expresses this beta-glucuronidase enzyme. A lot of us are familiar with this to an extent. Some of the stool tests on the market actually measure beta-glucuronidase activity, such as the gut zoomer from Vibrant Wellness, which is one of the stool tests that I'll use sometimes to take a closer look at gut health. But that beta-glucuronidase activity, which is really coming from the population of the estrobolome, well, its job is to basically recycle estrogen or estrogen-like compounds back into the body, right? So after we methylate estrogen and we conjugate it in the liver, then we're going to dump it into the bile, squirt that into the intestines, and ideally we're going to defecate out some of these conjugated estrogen metabolites. Now, beta-glucuronidase is going to deconjugate that estrogen, and then it allows it to be free and break loose, and it can recycle back through, be absorbed through the intestines, go back through the hepatic portal system, and contribute to higher levels of circulating estrogen. Now, this is not always a bad thing. I want to make this very, very clear. I think we get too binary. We try to label things as good or bad, and it's like the physiological clinical context is what determines that. So for example, if you have somebody that has, endometriosis or maybe breast cancer in a more severe circumstance, well, a higher degree of estrobolome activity and beta glucuronidase activity would probably not be a good thing because it's going to add more estrogen to an already overburdened estrogenic system that has a disease state of endometriosis or fibroids or hormone sensitive cancer like breast cancer, and it could be making that worse. But in other contexts, such as let's say more of an estrogen deficiency or even like a post-menopause, well, higher beta glucuronidase activity might not be a bad thing because there's not really very much circulating estrogen, but the beta glucuronidase is allowing more recycling of estrogen. So it's all about what are the levels of estrogen and estrogen metabolites and how is that influencing the clinical presentation and in context. But why I bring this in here is because the estrobolome is a major factor to consider with how much estrogen do we have circulating in the body, right? If we have a slow COMT and we can't really break down estrogen very quickly through that methylation reaction of our estrogen metabolites, and let's say on top of that, we have an estrobolome overgrowth within our microbiota, and we can use things like the Biome Effects from Microbiome Labs or the Gut Zoomer from Vibrant to get more insight into the composition of the microbiome and the level of beta glucuronidase enzyme activity. So certainly then, as if we have that excess of estrobolome beta glucuronidase and we have a slow COMT and estrogen is building up more and more and more, that then, like I said before, that high estrogen is actually going to down-regulate the expression of the COMT enzyme to begin with. Again, as you can see, this can be a little bit of a vicious cycle, right? So it's all about how do we support the genetic blueprint? How do we support the epigenetics? How do we modulate the hormone levels, the estrobolome or gut activity? There's a lot of layers, which is why I'm so passionate about functional medicine is we don't rely on like, well, this drug with a single mechanism of action, monotherapy, and that's supposed to correct all these dysfunctional pathways and enzymes. No, a holistic approach. We're working on a lot of mechanisms. We're working on modulating a lot of pathways for the better, right? And we get better results, generally speaking, when we work in that way, which is why functional medicine is so special. So just really highlighting that importance of the gut health aspect. I could keep going on that note, but we have a lot more to get through, and I'm just keeping an eye on the time. So now kind of drawing this a little bit more to mast cell activation syndrome, which has become such a hot topic, and I have a lot to say about the topic. So we'll sprinkle on, some clinical nuggets of wisdom here, hopefully. Now, this is really cool. So I think most of us can appreciate COMT does not metabolize histamine directly, but there is an indirect connection here. So for example, one of the major clinical pearls I wanted to share with you guys, I think we've made it clear that COMT plays a critical role with estrogen metabolism. Now, estradiol, E2, our major, most potent bioactive form of estrogen, is actually a very potent activator of mast cells. So if we have a man or a woman, it doesn't matter if they're a man or a woman, but if they have excess estradiol, this can contribute to mast cell instability, which then could potentially lead to more mast cell activation and degranulation, where then it's dumping all those pro-inflammatory mediators, the pro-inflammatory cytokines, the histamine, the tryptase, so on and so forth, and driving that sort of mast cell activation syndrome or symptoms of histamine intolerance, right? Now, we know that histamine is really broken down extracellularly by diamine oxidase. As estrogen goes up, not only does COMT enzyme expression go down, but so too does diamine oxidase levels and activity. Now, histamine methyltransferase, or HNMT, is the intracellular enzyme that breaks down histamine within the cells intracellularly. Now, of course, it's a methyltransferase enzyme, so we have to have adequate methylation and SAMe activity and biosynthesis to fuel that. But imagine if we have this slow COMT, high estrogen, high estrobolome activity. now we're also decreasing the ability to break down histamine and potentially having methylation issues here. So then as that estrogen creeps up and up, it's stimulating more mast cell degranulation, more histamine dumping, while also decreasing the ability to clear histamine through HNMT and DAO. So while a COMT polymorphism is not going to directly cause mast cell activation syndrome or histamine intolerance, it is indirectly contributing by way of estrogen metabolism in particular. Now, just a few other clinical pearls there. Testosterone is actually a major mast cell stabilizer. A lot of times when we're talking about mast cell activation syndrome in this industry, everybody just immediately associates it with mold and mycotoxins because that's just what people are used to hearing. It's so much more than that. And I do have a brand new ebook that I'm going to share with you all at the end of this presentation because there's more layers to the mast cell conversation than just mold. So testosterone is a major mast cell stabilizer, as is progesterone. So if you have somebody, again, man or woman, we're inclined to think of women when we think about estrogen and progesterone. But if you have a man or woman that has low testosterone with high estrogen, and especially if they also have low or absent progesterone, thinking about like polycystic ovarian syndrome, which is rampant in young women today, it's appalling. This can very much contribute to this mast cell activation, histamine intolerance sequelae and working picture here. So I have some good notes here. And of course, when we consider the hormonal aspect of it, but then also that dysbiotic leaky gut that's contributing to this picture. And then, toxic burden. What if there is mold exposure? What if there is, EMF hypersensitivity or other factors, nutrient insufficiencies, other genetic predispositions that might just be compounding this vicious cycle? So this is a great diagram that I created to illustrate the role of mast cell activation and excess histamine, because we actually have histamine receptors on the blood-brain barrier, and if they're hyperstimulated, it can cause hyperpermeability or leaky brain, so to speak. Our glial system, which my whole career literally centers around the glial system of the brain, or neuroimmunology here. So we do have histamine receptors on various brain cells, including our astrocytes, which maintain a healthy blood-brain barrier, and our microglial cells, my favorite cell of the human body. So mast cell activation can very much stimulate neuroinflammation and leaky brain as well. The mast cells in a lot of ways are kind of like the first line responders of the human body. It's really like these mast cells are what are responding to environmental input signals. We have a lot of mast cells in the skin, in the mucosal membranes, are different compartments of the body that are more external facing towards external foreign invaders and stimuli, which is why before mast cell activation syndrome was ever a thing, it was really just thought of as allergies. You're exposed to an allergen and you have a histaminergic type of response, anaphylactic response. And now we've evolved our thinking, and we have a more intricate understanding of these sentinel mast cells that do respond heavily to environmental input signals. But then, of course, we can't just hyper-focus on one type of immune cell, because all immune cells are designed to be activated depending on what's going on within the environment or the milieu that they're residing in. But I'm really trying to highlight this interface of the mast cell activation and how this also then drives glial activation and neuroinflammation within the central nervous system when the astrocytes and microglia become activated as well. So it's kind of one of those mast cell activation syndrome could drive what I refer to as microglial activation syndrome. So this is my favorite diagram I think I've ever created, and really my whole career centers around this. So then everything that I study is in relation to how does it affect this? So if we have those mast cells or macrophages that are being activated, that's going to drive this leaky brain effect, this hyperpermeable blood-brain barrier, and activate this neuroinflammatory cytokine storm within the brain. And neuroinflammation is a thing, it's a fire that keeps itself going. So for example, when our immune cells within the brain, the microglia, are activated into their pro-inflammatory M1 phenotype, and they're releasing all these pro-inflammatory mediators that can cause damage to our brain cells, like our oligodendrocytes and neurons. Well, as these cells are becoming damaged, they're releasing damage-associated molecular patterns that then bind onto pattern recognition receptors on the microglia to further activate them. So it's like a fire that's keeping itself going and fueling itself. And there are a lot of biomarkers that we can use to get more objective insight into the degree of immune activation, mast activation, glial activation, blood-brain barrier dysfunction, neuron damage, so on and so forth. Now, certainly when we have this neuroinflammatory storm going on, it does directly dysregulate our neurotransmitters. And just for the sake of time, we don't have a ton of time to go into all the cool biochemistry, but there's two major pathways: the kynurenine pathway, which is what is pictured here, as well as the biopterin-neopterin pathway, which is regulated by the GTPCH1 enzyme, and then we've got the IDO1 enzyme. The important take home to understand here conceptually is that when we have this neuroinflammation activity in the brain, it dysregulates our ability to biosynthesize some of our feel-good neurotransmitters like serotonin or dopamine or GABA, and we actually get too much glutamate, which can be excitotoxic and damaging, as well as other noxious mediators such as quinolinic acid or neopterin, for example. So this is my microglial activation syndrome graphic that's just highlighting some of the major root cause factors that can contribute to neuroinflammation and glial activation in the central nervous system. And this is where we have to be very objective practitioners because, for one client or patient, it might be things like hyperglycemia and insulin resistance and obesity, or maybe some anemia causing a bit of hypoxia. For somebody else, it might be they just had a concussion and hit their noggin. So now they have a glial activation and neuroimmune response in response to the head injury. For somebody else, they might be living in mold. Somebody else, they got bit by a tick 10 years ago, right? So we can't be biased in what our root causes are. We have to be impartial and unbiased and come into a case to really determine what root cause factors are relevant for this client or patient, and all the while making sure that we emphasize the foundations. The foundations of a healthy lifestyle are what pull the most weight, right? So moving this along, so I don't get on a soapbox here, I designed my own lab test called the mental map, which MAP stands for microglial activation profile. And so this is the main panel that I use in my practice because I really designed it to be the best panel to assess for leaky brain, neuroinflammation, and glial activation, but also deeply assesses neurotransmitter balance, methylation and epigenetic health, oxidative stress, and so much more. So I like to use my panel, the mental map, in conjunction with the MaxGen genetic panel, as well as looking at other functional labs as needed to really get a more clear understanding of what's going on with each client that I work with. Now, looking at the methylation connection here, because this is pretty critical. So this is actually that biopterin-neopterin pathway that I was just referring to as one of those biochemical pathways that becomes dysregulated. This pathway is really going to be where we produce our nitric oxide, our dopamine, our serotonin. And so essentially, if we have immune activation in an inflammatory immunological event, such as lipopolysaccharide-induced endotoxemia from a leaky dysbiotic gut as probably the best example, well, when we have a lot of these noxious pathogen-associated molecular patterns, such as LPS or mycotoxins or exotoxins or whatever it might be, it's actually going to dysregulate these enzymes, and we have a depletion of BH4 or biopterin. BH4 is a critical cofactor to be able to produce our miracle molecule, nitric oxide, our serotonin, and our dopamine. So as a clinical pearl, certainly you could look at certain SNPs on the MaxGen panel, obviously MTHFR, also methionine synthetase, BHMT, CBS, MAT. There's a number, pretty much all of the methylation related SNPs and enzymes are going to be directly pertinent here. But if you were to look at blood work and see that they have a high level of homocysteine, so hyperhomocystinemia, high levels of homocysteine directly imply that their biopterin is depleted. And then especially if you measure neopterin, which is on the mental map in the blood. So if you have a high neopterin and a high homocysteine in the blood, you can pretty much assume that their biopterin is depleted, which is going to decrease their ability to produce that dopamine to begin with. So for example, imagine if you have high homocysteine, high neopterin in the blood, and genetically they are more of a fast COMT. They break down catecholamines quickly. That person's going to be so apathetic and depressed because they don't have the biochemical cofactors to make the neurotransmitters very well, and they have a lot of maybe inflammatory pathways being activated. Meanwhile, their genetics predispose them to breaking down the catecholamines very quickly. So it's a very dynamic interface that we're working with in real time. And this is why, I mean, as much as I love lab testing, we're only going to, pull some blood or do lab testing, every 3-6 months, maybe. So we have to be able to recognize these things in real time to help them adjust their supplement protocol or adjust their holistic regimen to fine tune some of the ups and downs that they might be experiencing. We have to move really slow, titrate very slowly, especially in more delicate cases. And especially when we're working with mental health disorders, neuropsychiatric conditions, somebody's cognition and their mental health and perceived cognitive capacity, I think is a really sensitive indicator of what's going on biochemically, because you can feel very different day to day depending on what you're putting into the system and what you're upregulating and what you're downregulating here. So obviously we have to make sure that their methylation pathways are working properly and making sure that we're feeding their cells in their bodies with adequate levels of, folinic acid or methylfolate, the cobalamin, zinc and B6 don't get talked about enough in that conversation, magnesium, so on and so forth. So here's just a basic diagram of the methylation cycle. We don't have time to, go through methylation 101 for now, but very simplistic diagram that just illustrates that ability to methylate the folate, be able to produce S-adenosylmethionine. And it is that SAMe that is the cofactor to be able to really facilitate the COMT methyltransferase activity. COMT doesn't really do us a whole lot of good if we don't have adequate SAMe. So if we have high levels of homocysteine, the chances that they have adequate SAMe bioavailability is very, very low. And there's a lot of things we can do to support this. For example, creatine, for some odd reason, creatine's all the rage right now. And I love creatine. I've been taking it for like 15 years. Even just by taking 5 grams of 2 to 5 grams of creatine monohydrate daily, that can actually preserve a lot of SAMe because it takes a lot of SAM to manufacture creatine and biosynthesize it. So if you're replacing it with exogenous supplementation, it just frees up more SAMe that we can use to, break down estrogen or dopamine or norepinephrine or whatever. And especially if we have faulty methylation and hyperhomocystinemia, high levels of homocysteine, that can also contribute to leaky brain, excitotoxicity, and even potentially neuroinflammation through activating NMDA receptors on the blood-brain barrier or even neurons themselves. Probably don't have time to go into a bunch of nitric oxide stuff, but that biopterin is a critical cofactor to be able to produce nitric oxide, which has so many critical roles in the body, everything from blood flow and vasodilation and perfusion, lymphatic dilation for lymphatic drainage, or even glymphatic activity is really regulated by nitric oxide. Clinical pearl aquaporin 4, which is basically a water pipe that's on the end feet of astrocytes that wrap around the blood-brain barrier to transmit fluid between our cerebrospinal fluid and interstitial fluid. That's actually regulated by nitric oxide. So if we're talking about our ability to get toxins like tau proteins and beta amyloid out of the brain through the glymphatic and lymphatic system, nitric oxide plays a critical role. It also regulates a lot of our neurotransmitters and so much more. So maybe check out the lecture I did for MaxGen last year, because we went a little bit deeper into the nitric oxide interface, but just worth touching on as part of this proponent. All right, so moving this along, I think we've covered this relatively well. We talked a lot about the estrobolome and the microbiome interface. So really what I'm highlighting here is how if we have a dysbiotic, low diversity, Leaky gut, we've got the mucosal barrier is damaged, the intestinal lining is damaged and leaky. So this allows lipopolysaccharide, which can actually directly inhibit methylation, by the way, and drive a lot of inflammation. And then some of those pro-inflammatory cytokines will downregulate the expression of COMT, just thinking through some of the mechanisms here. So if we do have this leaky dysbiotic gut, it becomes such a critical component. It's really hard, if not impossible, to optimize or correct the neurochemistry if we don't address the gut. So certainly whole lecture and conversation about the gut-brain axis, but we touched on the role of the estrobolome with the beta-glucuronidase as well. So I like to use a lot of serum bovine immunoglobulins, prebiotics, spore biotics, all sorts of different nutrients, things like zinc, carnosine, even folate actually is pretty important to regenerate the enterocytes. And by supporting methylation, we can support the healing of the intestines, but also by supporting the gut health, we can also improve methylation. So why not do both is kind of my theory. So this is a schematic diagram showing that endotoxemia that I have been alluding to, because those lipopolysaccharide will directly provocate mast cells, macrophages, neutrophils, microglial cells, if they make it up into the brain, even through this leaky gut, leaky brain sequelae. So this is a little bit of our wrap-up slide. I recognize I'm already kind of pushing the time limit a little bit, but we're almost there, so we might as well power through. But this is a good slide to somewhat summarize a lot of the ground that we've covered. I know it's actually been quite a bit of ground, and certainly this is going to be more of like a framework to consider when you're approaching looking at your client's MaxGen lab report and then combining that with your functional lab testing to really determine, all right, what is the holistic protocol and program that I'm going to put them on? Some different considerations here. So somebody with more of a fast COMT, supporting dopamine retention, stress resilience, estrogen metabolism, whereas somebody with slow COMT, we need to be thinking about methylation and estrogen detox or histamine clearance and mast cell activation because of some of those mechanisms that we looked at. So slow COMT is generally going to need more of those methyl donors and methyl nutrients, the SAMe, B12, folinic acid, or methylfolate, magnesium, various polyphenols to an extent. Fast COMT is going to do better with some of those dopamine precursors like tyrosine or L-dopa, mucuna, along with different adaptogens or stress modulation. So by using the genetic testing with functional lab testing, we can really kind of further hone in their protocol. So as an example here, if you had a patient with slow COMT, plus they have estrogen dominance and high circulating estrogen, and they have high circulating histamine, I like to use whole blood histamine to quantify that. So then we would really want to be looking at, well, how do we support their methylation? How do we optimize their microbiome diversity and gut health and try to bring down the inflammation to get this pathway a little bit more back online? So some conclusion and key takeaways here. I'll let you guys read through this later, but I really just want to very quickly highlight this case study to give you a real life example of what this could look like in real life, because, it's easy to give a lecture and have it be like, wow, that was so cool, but how do we actually implement this and integrate this information into a real life case that can kind of get messy and blurry at times of we don't always know exactly what's going on, but this is where the clinical critical thinking really comes in. So this client, I'll make this really short and sweet, just highlighting the genetic component here, because this is kind of your, I want to say, prototypical complex chronic illness picture. I think it's one of those, she thinks she's more complex than she actually is. And I've kind of started getting a little bit of a intuitive red flag vibe when people claim to be complex. And I do think at times there might be a little bit of a layer of overcomplicating healing as a subconscious form of self-sabotage. Could it be that they have some self-limiting belief systems that they haven't dismantled yet, and they're making themselves out to be more complicated than it really needs to be? And this is where foundations rule the day. But once we have solid foundations in place, we can further bio-individualize with more precision based on genetics and functional lab testing. Now, this is the client I was referring to that she has OCD, and I will just directly, candidly, succinctly say, The OCD is the main problem. It's the main healing opportunity in this case. Is there a history of mold illness? Yes. Is there a history of medical trauma? Yes. Is there toxic burden? Yes. Is there metabolic dysfunction? Yes. Are there hormonal issues? Yes. The OCD, though, is the major obstacle to this woman having a healthier, happier quality of life. And that's really what we're focused on. Now, why I make such a strong point of that is in part because we talked before about how with the COMT, here's that Val 158 met single nucleotide polymorphism that this whole presentation was built around. So she is a slow COMT, you know, it says right here, the warrior gene. Now, I'm not saying that she has OCD in rumination because of this one SNP. What I'm saying is this particularly very well studied, clinically relevant polymorphism, it can be directly fueling some of her OCD symptomatology, right? Now, maybe, like, I would be really curious, and this is what research will need to answer in time. Some of these, you know, diagnostic labels that we receive, ADHD for me and major depressive disorder, OCD for her, are these diagnostic labels, maybe they're just a label that's really being driven by genetic predisposition, epigenetic modification, right? Because if I personally were to redo a clinical psychology or psychiatric evaluation, I wouldn't fit the criteria for major depressive disorder anymore. ADHD, maybe, it's a little questionable, but not so much depression. You know, I'm not a severely depressed person anymore. I didn't change my genes, I changed my epigenetics, right? So this is why I love the power of genomic precision medicine, is we can look at the genetic blueprint and say, you know what? It's okay that you've had chronic struggles with these issues. We have more clarity as to why that might be. And here's how we can better support you down to a genomic level. And so she's actually really into this information. And Bobby, who's here with us and works for MaxGen, she actually coached this client for like, I don't know, well over a year. So she could tell you all about it. But I thought this was really interesting. Now, I do want to also highlight before I wrap up here, of course, we can't just look at a single gene in isolation. We can't just look at a single SNP in isolation. So I did include some of her MTHFR polymorphisms just because it's relevant to the conversation, as well as CBS. So as you can see, she does have some other polymorphisms within, like H&MT that breaks down histamine, or CBS, or MTHFR, or MTR, that could be then further contributing to a lot of these mechanistic issues and dysfunctions that we talked about today. So with that, just for the sake of time, I really highlighted the main thing with the case study that I wanted to. So I have some reference slides that you can check out. This is just some of her mental map biomarkers that we've been tracking for literally years. So you can kind of see what this looks like when you're tracking the physiology over time. And then I just had a reference slide here. These are just some of my favorite therapeutic supplements to start a lot of my clients with. Obviously, I individualize it, but these are just some of the things that I find myself reaching for and recommending pretty often in a lot of cases. Very therapeutic supplements I think a lot of people could benefit from. And then finally, I like to emphasize, I really believe we have to spend more time tracking physiology than chasing root causes, especially when you have the warrior gene or OCD or a panic disorder. Chasing root causes just fuels fear and anxiety and stress. Tracking physiology is how we objectively know if we're moving the needle on particular physiological systems and actually getting clinical improvement. So with that, finally, I'll leave this up on the screen. This is my brand new MCAS ebook. I'm very proud of it. It's like 53 pages, 70 references, everything you could ever want to know about mast cell activation syndrome. It's 20 bucks, and the best part is a portion of proceeds actually gets donated to my not-for-profit research and education foundation to fund research for mental health. So it's supporting a good cause, and you could learn everything you would want to know. But with that, I'll go ahead and be done and hand it back to Chris.
Brendan, that was fantastic. Thank you so much. So informative, so much actionable stuff there. We do have some questions if you've got a few minutes.
Yeah, I don't have a hard stop, so happy to, yeah.
Great. The first and most obvious one is, will we have access to the slides? We meaning the registrants or participants. They're wondering if they're going to have access, or is that something that they would get through scanning the QR code and accessing the ebook?
No, yeah, I'll make sure to get you guys a copy of the PDF so you can send that out to the registration list.
Great, great. Then I'm going to get into a couple of these questions and we'll just kind of work our way through as best we can.
Yeah.
Someone says, I have COMT RS 4633 TC downregulated and COMT RS 4680 AG downregulated, slow COMT. I started experiencing anxiety age 10 to 11 years to the degree that by age 34 couldn't leave the house, which I managed to resolve myself. Early menopause and more recently autoimmune thyroiditis, assuming this is all poor methylation pathways combined with MTHFR RS 180 1133 question mark. There's a lot there. So
Yeah, I followed it. It was very characteristic of a lot of the client inquiry emails that we get where it's a lot of info and trying to unpack it on the spot. Now, what I'll say though is, I definitely heard a number of more naughty SNPs that could be predisposing to some of these certain kind of biochemical predispositions in general. So something I'm really fervent on when it comes to functional medicine and root causes. I think it's easy to get a little bit biased with being tempted to kind of blame all of our health complaints, symptoms, and life problems on like one specific kind of root cause or maybe a cluster, but there could be a lot of layers going on there. So that's where, giving a lecture like this, where we're going into a lot of specific pathways or mechanisms. But if I was consulting with this individual privately, the very first thing I would do is like, let's back up and zoom out at a very, very high level, looking at what are the symptom clusters, the medical history, the perceived quality of life, and really looking at psychology and lifestyle first and making sure foundations are in place before then zooming in a little bit of like, well, we have genetic predispositions for sure, but what's going on epigenetically and physiologically right now? Let's get some fresh blood work, some fresh lab testing so we know what's going on under the hood right here, right now. And then let's start titrating in certain bio-individualized nutrients slowly to see what's going to move the specific symptom. So it goes from this tons of information kind of vague down to let's get really, really clear and structured with what are we trying to accomplish and where do we start and what's the physiological baseline that we're starting with.
great. Another question, what does DHFR heterozygous, a 19 base point sequence of DHFR gene, is deleted in one copy of the gene associated with 2.4 times higher expression of DHFR? Is that question clear?
Yeah, essentially that would potentially lead to a deviance in the ability to convert folate into ultimately methylfolate. So a lot of times when you're looking at the folate pathway, like MTHFR always gets the press because that's, the famous SNP and enzyme at this point. I actually have these tank tops and t-shirts I printed that says Namaste MTHFR on the back, which I thought was funny, but it really comes down to being able to take folate that we ingest from the diet, particularly dark green leafy things. Now, it's worth also noting that our microbiota is a major source of de novo folate production. We actually have to be able to use the microbiome to convert folate that we get from our food into a bioavailable form of folate that our cells can actually use. So if we're talking about the DHFR enzyme, which is an intermediate enzyme within the folate pathway, I mean, first that folate has to go, into your mouth, through the gut, through the microbiome, be transformed into a bioavailable form, uptaken by the cells through folate receptors, and then converted through DHFR into a more bioavailable like tetrahydrofolate. So this is where if the sort of genetic weak link could be in part the DHFR, we can supplement with folinic acid or methylfolate to essentially bypass that in general. So it's kind of similar to the MTHFR rhetoric where it's like, well, if we supplement with the bioactive form, then we don't have to rely on our sort of faulty enzymes to do it for us. So I do actually tend to use folinic acid more than methylfolate with clients. It usually goes better in general.
Okay, great. Here's another one. I was exposed to folic acid, contrast dye, and nitrous oxide before I knew I had methylation problems. In 2022, after surgery, my body spiraled into an inflammation state. And now I'm all B vitamin and mineral deficient with malabsorption issues, experiencing nerve damage, I suspect due to the folate and B12 deficiency. How do I deal with this?
A lot of personalized kind of medical questions going on here. So certainly a little bit of a medical disclaimer, but
Of course.
offering just some insight there. Now, folic acid, and Ben Lynch is obviously very adamant about this, although there's a nuance to it. Again, with pop culture, we love to polarize and demonize things. It's all bad or all good. Now, don't get me wrong. I'm not a fan of folic acid. I don't think synthetic folic acid is the best supplemental form of vitamin B9. However, when you look at how they added a lot of folic acid and synthetic vitamins to cereal grains and processed food, it did actually decrease the occurrence of neural tube defects, which was the original purpose of all of that. Now, with that said, the problem with folic acid, it has a high affinity for folate receptors, but low bioavailability. It can lead to what we call an increase in unmetabolized folate. So it is better to supplement with folinic acid. Now, also, though, I can't emphasize this enough, optimizing microbiome diversity, because that really is the gatekeeper of your nutrient bioavailability, including folate and the production of de novo folate from some of your gut microbes in general. But using the exogenous folinic acid, methylfolate. Now, the nitrous oxide, the laughing gas, and everybody confuses it with nitric oxide. That second nitrogen makes a big difference, though. Nitrous oxide is laughing gas. Nitric oxide we all produce in our bodies. But that nitrous oxide can inhibit methylation temporarily. But, we're talking like you get dosed up with laughing gas for like oral surgery. You might feel pretty lousy for a few days, especially if you have impaired methylation, but it wouldn't really last chronically. Once you replete the cobalamin and the folate and get the methylation back online, because like if you measured homocysteine before nitrous oxide use and after, you would see an acute increase in homocysteine. But once you get it back down, you've corrected the methylation biochemistry. So to wrap that up, something I'll say, because a lot of these questions are a little bit hypothetical where we don't have the biomarker data to know exactly what's going on objectively. And so I just want to emphasize that, again, test don't guess of we don't want to create narratives in our head of we're still suffering from something that maybe we're not. Let's make sure that we're using the correct biomarkers to know, are we still B12 deficient, methylmalonic acid? Are we still hypomethylating, high homocysteine, so on and so forth. So that way we can be a little bit more precise with our intervention strategies.
Great. Another one here, if you've got time. I have slow COMT, high risk of H. pylori / yeast gut infection, histamine issues, and had an estrogen dominant cancer. Who do I go to be monitored or treated? Maybe.
I have a pretty extensive network in the functional medicine space. I feel like I'm connected to some of the best practitioners and brightest minds out there. I can't say any one individual comes to mind for that picture. I mean, certainly, my practice is always taking clients. You could always email info@metabolicsolutionslc to see.com if you want to see if my practice is the right fit. I have some great practitioners on staff. We're well equipped to help individuals that believe they're struggling from something really complex. And we have a good system in place to make it less complex, more actionable. Let us worry about the intricate details and let them worry about the behaviors needed to be practiced to really implement healing and achieve their goals.
Great. If folinic, gives more anxiety, slow COMT, MTHFR, etc., what could be the missing ingredient to help the pathway? What's your favorite supplement for brain inflammation?
Those are good questions. So for one thing, if somebody has more of a slow COMT, I'm not going to be pushing the methylated nutrients quite as much. And this is part of why I alluded to like the rationale. Sometimes I wish I could like plug my brain into your guys's so you could kind of see the thought process behind it. But in general, of course, when I'm making recommendations, I'm basing it off all available data, which might be a ton of blood work and maybe stool testing, hormone testing, genetic testing, history, symptoms, but also, preferences, like what is the intervention that the client wants to do? Like some people want to do psychedelics or peptides or tons of supplements or lifestyle. So, the program has to be catered to what's going to be palatable and integrated by the individual that's trying to heal in general. But specifically there, if they are more slow comped, I'm not going to be pushing methyl donors right out of the gate. This is where maybe like the creatine would be a good example, where creatine itself is going to improve mitochondrial function, lean mass, metabolic function, improve the bioenergetics of the brain, while also sparing some of our methyl donors. So to me, it's a more gentle way of promoting methylation without pushing a ton of methylfolate and methylcobalamin, because it is when you start, and to back up a little bit, this has become a problem where the methylated B vitamins have become the standard because of the prevalence of MTHFR. But the methylated B vitamins can be too stimulating for a lot of people. And a lot of times they're dosed too high. For example, it's become routine practice in conventional medicine where they'll screen people for the MTHFR polymorphism without looking at homocysteine or physiological biomarkers. And if they have the SNP, they'll immediately prescribe like megadoses of methylfolate, methylcobalamin, like 15 milligrams. To put that in perspective, most multivitamins will have like 400 micrograms of methylfolate and methylcobalamin. That's like a starting dose, but most methyl support, it's starting at like a milligram, 3 milligrams. So I think with methylated B vitamins, you want to start really low and titrate them up slowly. And if you're mixing too many things, like let's say you pop, a gram and a half of tyrosine and you pop, 2 milligrams of methylfolate, methylcobalamin, that's probably going to cause anxiety in a lot of people, regardless of COMT status. So there's definitely an art to the titration and bioindividualization.
Great, great. How about this? Can you elaborate on why folinic acid is preferential to methylfolate? Is it because pushing too many methylated nutrients can be harmful for people struggling to use slash excrete it downstream, as discussed today?
Yeah, there's a few interesting layers to that. For one thing, I'm thinking of some of the research I cited in my methylation course. There are some studies that postulate that folinic acid might be the more superior form of folate to supplement with for a number of reasons. And folinic acid, as opposed to methylfolate, folinic acid is going to promote DNA production more effectively. Methylfolate skips the SHMT pathway in general, which is where we get some of our nucleobases to actually create new DNA for new cells. So especially like as a clinical pearl, anytime I'm working with like a expecting mother or a mother to be, they're literally growing a human baby inside of them. That takes a ton of cell replication, which requires a ton of new DNA production. So folinic acid is really the superior form to promote that DNA stability and DNA synthesis for new cells. And especially if we're healing from chronic inflammatory disease, there's a lot of old damaged cells. So the folinic acid is really promoting the DNA replication on top of being less stimulating. So kind of lower side effect profile with potentially other biological benefits.
Okay. Couple more if you have time.
Yeah,
Uh,
I'm good.
what is your quick solution for someone with or really struggling with sleep issues? That might be a tough one to tackle easily, but
Yeah, yeah. I mean, the funny thing, my line of work, it's, I'm kind of equal parts practitioner, clinical educator, and then clinical researcher, and trying to like switch gears because the language and the thought processing is a little bit different. Because if I put on my practitioner hat, it's really more about practicality, right? We can't talk in ideals and, there's a meta-analysis to prove that this is the way to go. We're dealing with a real person in real life. And a lot of times we're not going to always have all the lab testing and all the data that we want to feel more confident in our recommendations. So, I mean, this is what we call empirical intervention. We're using intuition, we're using clinical critical thinking to decide what would be a good starting point, right? And especially with healthy coaching psychology, which makes receiving recommendations a lot more palatable and empowering, I'm going to be motivational interviewing of like, okay, so you have sleep issues. Why do you think you have sleep issues? What factors do you think could be contributing to those sleep issues? What do you think is something that you could do to try to improve your sleep hygiene and your sleep issues right now? Do you want to take a few supplements? Here's some supplements that I like to take for sleep issues. Have you tried them? Do they work for you? So rather than like, well, let me just spit out a singular recommendation as if it's going to be an end all be all panacea fix, let's do some critical thinking and motivational interviewing to see what's a good starting point to try to mitigate this. Let's make it action oriented. Now, I certainly, just for the sake of not being too cryptic or ambiguous here, there are a lot of sleep support supplements that I take every day to help me unwind. I love Bifidobacterium longum 1714, specifically ZenBiome from Microbiome Labs. That's a great stress reducer, changes your brain waves, helps with sleep. Or BDNF Essentials from Research Nutritionals is one I take every night. Theanine, melatonin, magnesium, bisglycinate. So many different soothing, calming herbs and supplements that you can take more allopathically while you're trying to optimize the sleep hygiene and the lifestyle routines and all of that to promote a deep restorative sleep at night.
Great. So lastly, this is kind of an amalgamation of a couple of questions, but what are your clinic locations, and do you offer telemedicine consults if physical visits aren't possible?
Yeah, great question. I'm all virtual actually. So maybe someday I'll have a brick and mortar, maybe, but the virtual life, it's a good life. It works pretty well. So we consult with clients all over America. International is a little bit more of a struggle. So if you email info@metabolicsolutions.llc.com, that's the easiest way to get in touch. I have a team of practitioners that work underneath me, and I personally oversee every single case in the practice, but I don't interface with the client directly anymore just because of limited bandwidth. So I let my practitioners really do the work, and I oversee and provide guidance as needed.
And would you repeat that email address?
Yeah, it's just info@metabolicsolutions.llc.com. I
Yeah.
think oh, there it was.
Okay.
There you go.
Last one, where can we get a tank top? These tank tops you talked about, they're blowing up here. Where do we get them?
Yeah, well, I do have I'm trying, it's been a minute. I think if you go to Instagram, there's a link in my profile bio, or it's on the main page of the website. But we do actually sell these tank tops and t-shirts to raise money for my research foundation. I can't say that they've been as hot of a seller as I would prefer, but we do have some merch that we need to move, and it does raise money for a good cause. So if you're inclined, you can snag one off the website.
I think we've got some people interested. So thank you
Awesome.
so much. We covered as many of the questions as we could. I hope everyone was gratified and satisfied with all of that. That was fantastic, by the way. I'm just going to take a quick minute to mention that going live today on Spotify, everybody be sure to tune in to the Truth Moms podcast, as our founder, Dr. Chad Yarborough, breaks down how genetic testing is revolutionizing personalized care. So don't miss this powerful conversation on episode 10, DNA Blueprint Decoding Genetic Testing. The link will be in the chat. And then next month's Practitioner of the Month, if you don't mind sitting in for this, Brendan, we're thrilled to spotlight Greer McGinnis, MS, RD, CLT. She's the next Practitioner of the Month in April for the link between autism and the toxic load. As a detox specialist, herbalist, and founder of Biomedical Healing for Kids, Greer empowers families with holistic solutions for autism, ADHD, and pediatric health. So we'll send out invite links via email to follow this event, and you can always follow us on social at MaxGen Labs. Thank you so much for your time and all of your brilliance. That was wonderful.
Thank you, Chris. I really appreciate you and MaxGen for having me. It was a lot of fun.
Let's do it again.
Awesome. Sounds good.
All right. Thanks to everyone who attended. We appreciate you. And we'll make sure to get these slides available to you.
Thank you all so much for attending. It was a good time.